Oh, it looks like I think we have the BC4031. Oh, shoot. That's what happened. Sorry, let me go back. So it looks like people are struggling with the exam, so this needs to be deactivated. Sorry about that. I just came out of that one. Let me come here and activate this one. Activate that. And now hopefully the exam 4 Q&A for BC351 has popped up. Is that okay? Yep. Okay, so it looks like people are starting to populate it with questions. Okay, great. All right, I've got that fixed, so that's good. Thank you, Molly, for pointing that out. When are all the extra credit opportunities due? So you're going to be, so the only one that we really have left is going to be the chapter chapter questions. Okay, into chapter questions, the practice material questions in the book, and those are due the same time as the exam is done. Okay, so everything else is all those due dates have now passed, and so I'll be putting up those extra credit, what you got in terms of extra credit on Canvas over the next couple of days. So I think I've already put up what you had for the discussion forums. I know I did last week. So we still have the chapter and then the Q&As, and then we'll go from there. So, okay, all right. All right, can you go over the short answers on the practice exam? Yes, I can do that. So there's 5 people that have said that would be great. In fact, the next question is short answers on the practice exam, so that makes 8 total. So let's go ahead and look at that. Let me come here and make sure that I've stopped my video. Yes, I have. Okay. So short answer questions on the exam. So this is our practice exam. Okay. Oh, this one's a fun one, right? I mean, in his studies of alcoholic fermentation by yeast, Louis Pasteur noticed that the sudden addition of oxygen to previously anaerobic culture of fermentating grape juice resulted in a dramatic decrease in the rate of glucose consumption. The effect has been called the Pasteur effect and can be counteracted by the addition of 2,4-dinitrophenol, or DNP, an uncoupler of oxidative phosphorylation. Why would the yeast cells consume less glucose in the presence of oxygen? Great question. Why would DNP counteract or prevent the Pasteur effect? Okay, so let's look at this. Why would they consume less oxygen in the presence of, less glucose in the presence of oxygen? So let me draw it this way, okay, and use this to this diagram to essentially answer the question. Okay, so what we have here is this, right? Glucose is going to be converted to 2-pyruvate. Okay, now we know that this is going to, and I'll draw up net here. What's going to happen net is going to make 2-ATP. Okay, and this is also going to make NADH, 2 total NADH. Now, let me do this. I'll do it kind of in the reverse of the way the question was asked, but let's do this in terms of if oxygen is present. So let's draw it. Oxygen is present, and we'll draw my prototypical version of the mitochondria here. Okay, better have the electron transport chain down here. Okay, so there we are, and draw this here. Okay, so we've got that going on here, and what's going to happen then is that this pyruvate is going to snake its way through the outer membrane into the matrix. So this would be the N side, this would be the P side, and this would be the cytosol. Okay, and in it goes, and now we have pyruvate here. 2 of them. Okay, and that pyruvate is going to be converted to, you all say, acetyl-CoA by that wonderful complex of enzymes called PDC. So acetyl-CoA, and that's going to make a small amount of NADH. Oops. Okay, and then acetyl-CoA we know is going to combine with oxaloacetate and it's going to become citrate. And this is going to be that wonderful thing that we call the citric acid cycle, and it's going to go round and round the loop it goes. Okay, I always like to draw it this way. Succinate is converted by complex 2 to fumarate. Okay, and this is converted, of course, through multiple steps, and we're skipping lots of steps here, but to oxaloacetate. Okay, and we know this, that these two acetyl-CoAs are going to lead to the production of 3-NADH from this process. Okay, the FADH2 is going to be up here, so I'll just go ahead and put 2 FADH2s here. And then finally to GTP or ATP, we'll just put 2 ATP. Okay, and what we essentially have here is that these NADH then here and here, okay, they're all going to combine, and even the NADH out here, although we won't discuss how it gets in here, but well, we have. I mean, it's the malate aspartate shuttle, or you know, one of those other shuttles, but it will get in here, right? And it will just essentially come here, and NADH then will give its electrons here, right, on the NAD plus, and I could draw up a lot of details here, but essentially this is going to make oxygen into water, or convert oxygen into water. Okay, and electrons are going to flow, right? And electrons will flow from here, and that's all going to happen. Okay, and then protons are going to be moved from N to P side. Okay, that would be the big thing here. Protons, zoom, protons, zoom, protons, zoom. And then all those protons are going to accumulate on this side, right? Lots of them, and they're going to go flying through here. And the really cool thing about this is that this is going to make ATP, a lot of it. Okay, so ADP plus PI. That makes ATP. Right? Awesome. Okay, and if we were to take everything that I've drawn up, okay, this whole process, and I'll write it in big pink here, is going to make anywhere between 30 to 32 ATP. And that's assuming a C ring of 8 subunits, okay, and a PO ratio that then comes out to be 2.5 ATP per NADH and 1.5 ATP per FADH2, okay? All right, now, that's with oxygen, okay? What happens if you don't have oxygen? All right, let me use yellow here. I'm going to get rid of this oxygen, going to get rid of this. All of this is lost, 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 right? All of this ATP is lost, and what then happens is that we're left with glycolysis to meet the cell's demands, and that's 2 ATP per glucose, and that's these guys right here, okay? Now, of course, to do that, this pyruvate is going to be converted to either lactate, or in the case of yeast, it's going to be carbon dioxide, okay, and ethanol. We'll deal with mammalian metabolism, but that's what's going to happen, okay? Now, notice that this is then 1/16 as efficient as the production of ATP if you have oxygen present. And so that means it's going to have to burn 16 times more glucose in order to meet ATP demands, right? So come up here, it says, in his studies of alcoholic fermentation by yeast, Louis Pasteur noted that the sudden addition of oxygen to previously anaerobic culture of fermentating grape juice, that's going to be our 2 ATP, okay, results in a dramatic decrease in glucose consumption, because when we add addition of oxygen, we get 32 ATP. Okay. And this is effect called the Pasteur effect and can be counteracted by 2,4-dinitrophenol, an uncoupler of oxidative phosphorylation. We'll get to that in just a second. Why would the yeast cells consume less glucose in the presence of oxygen? Well, because they don't need to burn as much glucose because they get 16 times more ATP from one glucose than if they do in anaerobic conditions. More than enough to meet the demands of the cell. Now, why would DNP counteract or prevent the Pasteur effect? Okay, so now we got to come here and we got to remove all our yellow stuff. I don't want that yellow guy there. Don't want this yellow guy here. Don't want this yellow guy here, right? All the stuff that we said, oh, this is not going to happen anymore. Okay, well, we're going to keep oxygen present. Okay, and there we go. And I erased a lot of other things, I know. Okay, I lost oxygen. Okay, there we go. Right, so all of this is going, and that's great, okay? But what it means by counteract the Pasteur effect is that even in the presence of oxygen, the cell has to consume significantly more glucose. That's what it means, okay? Well, here's what DNP does. Okay, DNP can hijack these protons and take them through the membrane independent of ATP synthase. And so it can diminish the proton gradient without making ATP. And so that means that instead of making 30 to 32 ATP per glucose molecule, it reduces down to, let's say, 16 ATP or 5 ATP, or who knows, right? It depends on how much ATP, it depends on how much DNP you have, but it'd be significantly less than the amount of glucose or the amount of ATP you can get per glucose if you have oxygen and no DNP. Okay, and so then DNP counteracts this or short circuits this process by removing the protons from the matrix, from the intermembrane space and puts them in the matrix independent of ATP production. So now the entire oxidative phosphorylation process decreases in efficiency. So, okay, all right. So that's the answer to, that was a short answer question. So that we did it. All right, so let's come back here. Can you go over the short answer? We got it. Okay. Inhibition of the Q cycle study guide questions. I don't have the study guide open, but I can open it. Go ahead and open it up. Get that up and I'll look at those. Inhibition of the study of Q cycle. Can you be more specific, maybe the person or persons that are asking that? Which question is it on the study guide that you're concerned with? Oh, I get it. Number 11. What if I inhibited the Q cycle? Ah, right. What would be the redox state of redox centers in complex 4, oxidized or reduced? What about the redox state of redox centers in complex 1 and 2? What would this do to the citric acid cycle and NADH levels? What about the production of ATP? Let me grab a drink of water and then I'll answer that question. Okay, so let's pull up another one of these things here. Boy, there's so much of this that's good on here. But let me go ahead and I'm gonna draw it up somewhere else, because if I do it on that, then I'll go real fast and people will be like, wait a second. So let's do this. Okay. Draw our boxes. Okay. Hopefully you recognize this. You say that is the high resolution structure of the electron transport chain. Awesome. Captain America shows off his artistic skills once again. Okay, so let's label these things here. 1, 2, 3, 4. Okay, and sometimes this is called complex 5. Oops, but I didn't draw a 5. There we go, got a 5 there. Okay, now we got it. All right, so what we're essentially saying is this, that complex 3 is being inhibited. And so what I want to do I think that I used this analogy before, but I'm going to go ahead and use it again because maybe you didn't hear it or you didn't connect with it at first, but I'm going to go ahead and say it now. Okay, so the analogy is this. If you were to think about the electron transport chain as a room, and that room has two front doors, okay? Complex 1 and complex 2 are the front doors, okay? And we're going to try and fill this room up and with electrons or people, okay? The second door is this. Is that complex 2. And it's going to get its people, quote unquote, electrons, right? From succinate, okay? And so these people or these electrons enter into door number 1 or door number 2, okay? And then they're going to leave through the back door, okay? And the back door is oxygen. And as long as the back door is open, then the front doors can continue to allow people to come through, or electrons to come through, okay? And the reason being that then electrons can leave and the room doesn't fill up, okay? That's the idea here. Okay, now in our last discussion, we had removed oxygen, and so we shut the back door. And when you shut the back door, then people or electrons keep coming through the front door, but they can't get out. And so pretty soon, then the room, if you think about just like this constant flow of people going through the room, out the back door, in through the front doors, right? Pretty soon that room will become filled up with people, and no people can no longer enter into the two front doors because the room's full, okay? And then those people, okay, coming from NADH and succinate, those things will start to fill up. They'll get larger and larger in concentration, and they'll feed back and inhibit the citric acid cycle, and you lose all mitochondrial metabolism, okay? All right, so now what's the idea here? Well, the idea here is this, in terms of the study guide question, is, well, what if you were to inhibit this process right here? You introduce a divider, okay, into our room. And that divider has no doors, right? So this would be the idea would be like a hotel lobby or like a hotel ballroom, right? And you've seen ballrooms that have these big dividers that you can open and close, okay? Well, that's what we'd essentially be doing here, is that we'd be taking that divider and be slamming it shut in the middle of the room, okay? So that all of a sudden the back door remains open, right? And you can just imagine all these people in this room, and then the divider gets put up. Well, there's all these people here, right, on this side of the divider that leave through the back door. They're like, I'm out of here, right? So they leave through the back door, but then there's all these people coming in that can't get through the divider, can't get to the back door. And so what happens then is this. Everything on this side, where I just put the star, will adopt, the room will be full, or they'll adopt the reduced state. So complex 1 will be reduced, complex 2 will be reduced, and then everything in complex 4 will be oxidized because all the people or electrons left through the back door, okay? All right, so that is essentially what will be happening in this scenario. So go back to our study guide question. Number 11, it says, it was number 11 on chapter 13. What if I inhibited the Q cycle? What would be the redox state of the centers in complex 4? They'd be oxidized. What about the redox state of the redox centers in complex 1 and 2? They'd be reduced. What would this do to citric acid cycle and NADH levels? NADH levels will rise, citric acid cycle flux will go down because NADH is a inhibitor, in some cases competitive inhibitor, of the enzymes found in the citric acid cycle. What about the production of ATP? Well, that's the only thing I didn't address. The production of ATP would go way down. And the reason is because the electron transport chain was shut down. And you can no longer then establish the proton gradient. If you can't establish a proton gradient, you can't use complex 5 any longer. So, okay, right. Awesome. Short answer on the practice, got it. Oh, summary of PDC. Yeah, so the 5 things about the PDC, that's what you kind of want to do here. And then we'll hopefully get to this next question too, because that one looks awesome. All right, so the PDC, let's see here. Really, you want to keep in mind the 5 things that it's going to do, the 5 steps. Okay, let me do it in this way. E1. Okay, it's going to decarboxylate pyruvate. That's what E1 does. Okay, now E2, if you remember, E2 is where our arm is. I'm not going to be able to draw this, right? So I'm not going to try and draw it, but maybe I'll pull up one of the lectures and we'll look at a drawing after I kind of write this out, okay? But essentially, there's a decarboxylation of pyruvate. That's the first thing that's going to happen, right? E2 is going to have that big arm, and there's going to be a redox step, okay? So it's going to essentially carry, E2 is going to carry an acetyl group to essentially the active site from E1. And you say, what active site? That would be the active site in E2. And then it's going to make, in that active site, acetyl-CoA. Okay. Now, E3 is going to then essentially make NADH. Okay. So those are the essentially the things that it's going to do. Now, you know, what you're kind of, I said 5 steps, what you're kind of missing here is that right in between these two, it really happens in E1, but E2 is involved, is a redox step that will essentially take the acetyl group off of E1 and put it onto E2, okay, right, which is this in between the two. Okay, you have to have both of them. But that is kind of a summary of this. Now, let me go ahead and pull this up because I think it would be helpful to look at it. Sorry, I have to find the lecture. There we go. 12. This will work. Okay. This is kind of the best summary of what I've got here. Okay, so I said, remember on step 1, I said that it would decarboxylate pyruvate. So that was what I said here, decarboxylate pyruvate. It's happening in E1. Come back up here. That's being shown right here. Okay, this guy right here. Okay, you have pyruvate, it's got this carbon on it. That carbon's going to come off, and it's going to become this hydroxyethyl TTP is what that's called. Okay, now step 2 I said is in between. There's this in between step. Well, that's this arm coming out of E2, and I didn't write this one out, but this arm is going to reach in here and it's going to carry out this redox step. This would be a redox step, and that's going to take that acetyl group off of E1 and put it onto the arm. Okay, so if I were to come up here, I thought this was it. Oops, there it is. And I could put right here, just in between these, redox step. It's kind of in between E1 and E2. It's happening in E1, so I mean you could put it in E1, but E2 has to be there for it to occur. Okay. Now, next, right? It's going to carry that acetyl group all the way over here into step 3, and it's going to make acetyl-CoA, and that's Ne2, right? And then steps 4 and 5 are kind of combined. I just said make NADH, okay? But that's going to happen all in E3, right? So when you come up here, I guess I went through that really fast. Let me come here. This acetyl group, let me erase a bunch of this stuff because it's kind of, there we go. So we got a redox step, right? This redox reaction here reduced this arm. So that's going to come all the way over here, and you're going to break off that acetyl group, okay? These carbons here will be broken off and make this acetyl-CoA, and the arm remains reduced, okay? And that's what these circles are showing. These SH groups are reduced. They have electrons on them. Well, they're going to come in steps 4 and 5. We didn't spend a lot of time on it, even in the original Echo 360 lecture. Steps 4 and 5 are going to take those electrons off of that. They'll pass it through an intermediate of FAD onto NAD plus, making NADH, and that will oxidize it. Okay, and so that's what I have coming back to here. Make NADH, and that would be steps 4 and 5. Okay, and this would be here steps. Let me just do it. Step 1. This would be step 2. This would be step 3, and this would be step 4 and 5. Okay. All right, so that's a summary of PDC. Okay, I expect you to know, essentially, kind of be conversant on that at the very minimum. Okay. Okay. Now let's go on to this next one. Got all these. I've got all these. All right, this one's cool. Can you explain why ATP synthase goes from O to T to L? That just seems backwards from what you want to happen. Well, it doesn't go from O to T to L. I'm wondering. So let's. When you look at an individual, let me think about this, how to address this question. It doesn't go OTT to L, it will go from T to O to L. So I'm not sure where people are getting that order. And I'm trying to remember because I've had this question before, and it was a confusion based off of a figure, and it was just a miscommunication. So it might be running into that miscommunication. But let's be clear here, okay? When we think about this, an alpha and beta pair, right, can be in the T, and if it's in the T that has ATP bound, okay, and then the O conformation, it'll go from T to O, right? This would be nothing bound. And then the last one would be, it goes to L, which is ADP inorganic phosphate bound. And then it will go from that back to T. Okay. And so each one of these little arrows that you have here, this would represent a 120-degree turn. Okay, of the gamma shaft, which is being turned by the C rings, right? So we go 120 degrees, you start in the T, well, that is gonna, right, 120-degree turn is gonna force it into O, and that will mean ATP comes out, okay? Right, this next 120 degrees means that you go from the open nothing bound to ADP and inorganic phosphate bound. Okay, ATP hasn't been made yet. Okay, this would be another 120 degrees, and we're back at T. And then it will go through the next 120 and goes to O. So it goes TOL, and not the order that's listed here, OTL. So maybe if someone wants to pipe in and clarify why they thought that it went from O to T to L, because I know that I've seen that miscommunication before, and I just can't remember where it was in one of the slides. But I want to be clear that it is.
There's a figure in the book that makes it appear like that.
Okay, maybe. Let me pull up chapter 13 and let's look at it.
It's figure 13.18.
13.
It shows an arrow that goes from O to T and then to L, which is why that's confusing.
Okay, let me pull that up. That's great. So it's figure 13.18. Is that what you said?
Yeah, that's correct.
I wonder if it's in this slide. Let's see if it's in these slides. Otherwise I have to pull up the book, which I don't really want to do, but I can if I have to.
It's slide 28.
Ah, okay, great. Slide 28 for students, which will be a little bit different for me. 13.18. This one, right? Is this the one we're talking about?
Yeah, that's it.
Okay. All right, so let me erase all this junk so we can look at this. Oh, I see. Okay, so I think I know why, because so what you're thinking here, O to T to L. Well, maybe I don't understand. Because what I'm looking at this figure, of course, I was the one that generated these figures. So what you have here is this gamma shaft that's got this point right here, right? And wherever that point is pointing, that alpha and beta dimer are in the O position, right? So if this cranks, and then this one next to it and 120 degrees adjacent to it is in the T, okay? So this is gonna crank, and that's gonna force this ATP out, and this will become O. This will become L, and this will become T in that 120 degrees, right? So we would draw, of course we can't, the blue arrow would become the pink arrow essentially now, because the gamma shaft has been shifted. Okay, and then we do, oops, and then we do it again, another 120 degree turn, and now it's pointing in this direction. Oops, draw a different color, and then this would become, right, O, this would become L, and this would become T. Does that clarify things for everyone? I hope so. Hopefully. Okay. Okay, so I'll have to think about that one. If you have questions about that, so looks like, yeah, that makes a lot more sense. Okay, good. Okay, great. All right, people are saying yes, that clarified things for them. Okay, so if you have more questions about that, certainly hang out afterwards. So summary of PDC, we've done that. Can you go through the last question on quiz 14, calculating 86 ATP? Yes, I can. Trying to think here. Can someone just tell me what size the lipid is, how many carbons it is? The person that asked this question, and then I can really help out. I mean, I can pull it up, but it'd be easier for me to just, okay, someone's got it in the chat maybe. Oh, there we go. 12, 7, okay, 16 carbons. Okay, that's what I needed to know. That's great. Wait, make sure you want to leave. You can minimize the window. I don't want to leave. No, that would be bad. There we go. Okay. All right, so let's come here. Let's come get a blank piece of paper. There we go. This will work. All right. There we go. So I'll just give you kind of the strategy on how to figure out a problem like this, okay? So it's 16 carbons, and I'll just do it the 16 carbon, but then you can do it with any number of carbons, okay? So what you got here is this. This would be essentially a fatty acid that's 16 colon 0, 0 meaning no double bonds, okay? And what you're essentially going to see is this. Oh, crap. Keep doing that. There we go. What you're essentially going to see is this. This is going to be converted to 2-acetyl-CoAs. I'm not 2-acetyl-CoAs. What am I talking about? Sorry. It's going to be converted to 8-acetyl-CoAs. There we go. Now, if you remember this, then your job becomes easier. Acetyl-CoA is 2 carbons long, okay? And so when we think about, like, well, how many acetyl-CoAs will a particular lipid produce? Well, in this class, okay, in anything really, actually, you can just say, well, I divide my total number of carbons by 16. I'm sorry, by 2, and that'll give me the total number of acetyl-CoAs I get, and that's 8. Okay. Now, the number of rounds that it was going to take to do that, right? Because it's going to, beta oxidation occurs by removing acetyl-CoA one at a time, and it takes 4 steps to do that. It takes a dehydrogenation step, a hydration step, another dehydrogenation step, and then finally the lysis step, or the thiolysis step, which is essentially breaking the acetyl-CoA off. Okay, and it has to keep doing that, and that would be those 4 steps are 1 round of beta oxidation. Okay, and so when you think about this, how many rounds will this take to get 8 acetyl-CoAs from 16 carbons? That number, the number of rounds, is always going to be essentially your total number of carbons divided by 2 minus 1. Okay, so this would take 7 turns to do this. And the reason it takes 7 is because the last molecule that's made, okay, is a 4-carbon molecule. And you take that last cleavage step will produce 2 acetyl-CoAs. Okay, so you always have 1 less round than you do total number of acetyl-CoAs, essentially. Okay, so this takes 7 rounds. Okay, now, because we're dealing with saturated fatty acid, each round produces 1 FADH2 and 1 NADH2. Okay, so this is going to make 7 FADH2s, and it's going to make 7 NADH. Okay, and that's beta oxidation. Now, the question asks you, well, how much ATP will it produce? Okay, and when we think about total number of ATP, well, then we have to think about acetyl-CoA, what it can be converted to. So acetyl-CoA can be converted to 16 carbon dioxides, and that's through the citric acid cycle. Okay, now the citric acid cycle will make 3 NADH, 1 FADH2, and 1 ATP per turn. Okay, and every time we put an acetyl-CoA into it, it'll turn 1 time. Okay, now the NADH come from isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase complex, and also malate dehydrogenase. Those 3 steps are going to make NADH. Okay, the FADH2 will be made by succinate dehydrogenase, and the ATP or GTP, whichever you want to say, will be made by succinyl-CoA synthetase. Right, and that's 1 turn. So this is to get, we're going to have 8 acetyl-CoAs. Each acetyl-CoA represents 1 turn of the citric acid cycle. So this is going to make then a total of 24 NADH, 8 FADH2, and finally 8 ATP. Okay, right. Now the question gives you a C ring, right, of a particular size. So I got to come back to this and look at this. A 12 subunit C ring. Awesome. Okay, because we have to calculate PO ratios. Okay, because these NADH, and there are 31 of them, and these FADH2s, and there are 15 of them, can be used to make a certain number of ATP depending on how much, depending on your PO ratios. Okay, so So we have another whole math problem. So let's leave that there, and we have to figure out our PO ratios for this, okay? So when we have a 12 subunit C ring, okay, for every 360-degree turn of the C ring, it will make 3 ATP, okay? The number of subunits is equal to the number of protons it takes to make a 360-degree turn. Okay, so if you have a 12 subunit C ring, and it will take 12 protons to make a 360-degree turn. So 12 subunits equals 12 protons, and I'll just put down here, oops, P to inside, okay? And this will equal 360-degree turn, which equals 3 ATP. Okay, now if you had, I mean, we could do the same thing, right? It would be 15 subunits, then it'd take 15 protons to make a 360-degree turn to make 3 ATP, right? If you had 9 subunits, then it would be, right, 9, 9, and 360 and 3, right? I mean, you can see how this goes, right? Right, but we're working with this one, okay? This guy, 12 subunits, 12 protons moving from P to inside to make a 360-degree turn to make 3 ATP. Now that means this. That means that if we take 12 divided by 3, it takes 4 protons to make, oops, that's a little bit too close. 4 protons to make 1 ATP, or 120-degree turn, right? This one would take, right, 15 divided by 3, it would take 5 protons, right? 9 divided by 3, it take 3 protons, right? So on and so forth. You get the idea, right? Now, we always have to do this, and this is where, you know, it can be, I mean, just even more fun. You always have to add plus 1 proton for pi, because we got to move pi into the matrix to make ATP. Okay, so this means this, when we add all this up, there's a total of 5 protons needed per ATP. Okay? Now, NADH moves 10 protons at a time. Okay? So NADH goes through complex 1, it goes through complex 3, and it goes through complex 4, right? And that will move a total of 10 protons, right? Those 10 protons then can be used to make a total of 2 ATP, right? 5 of them will be used to make 1 ATP. The other 5 of them will be used to make the next ATP. So in this scenario, when we have a 12 subunit C ring, right, we're going to have NADH is going to equal 2 ATP. Okay. All right. So that's for NADH. Now, when we think about it for FADH2, then FADH2 is going to move 6 protons, right? Because the protons go through complex 2 that doesn't pump any protons, and then it goes through complex 3, which pumps 4 protons, and then it goes through complex 4, which pumps 2 protons. Okay, so 6 protons total. 5 of those are going to make an ATP, right? Which isn't going to leave very much left over. You're going to have 1 ATP. That'll be 0.2 of an ATP. So we get then for each FADH2, we get 1.2 ATP. 6 divided by 5. Okay, and 5 again needed to make the ATP. Right. Now, what we can do from there is we can then essentially take all these numbers and multiply them, right? So you essentially take 15 times 1.2, and we would take 31 times 2, right? And we get our values, and then we would have to add in the 8 ATP plus 8 ATP. that are made directly through this whole process. Okay, and that will give you, let me just go ahead and real quick do that. 15 times 1.2 is equal to 18. Okay, and 31 times 2 is 62. Right, so that was pretty easy. So plus 8, 70 plus 18 is 88. And you all say, that's right. I put 88 and I got it wrong. Right? So what did I do wrong? I don't understand. Well, here's the thing. Okay. So this whole thing, when you add all this up, you get 88 ATP. However, there are 2 ATP that you have to subtract from this whole thing to give you 86. And the reason that you have to do that is because we don't actually start with, oops, sorry about that. We don't actually start with 16, 0 to make for beta oxidation. We have to start with a fatty acid that has had a coenzyme A added to it. Okay, and so we have to run the acyl-CoA synthetase reaction, and the acyl-CoA synthetase reaction will take a fatty acid and make a fatty acid, or what's called an acyl-CoA, out of it, and that will take 2 ATP. Okay, so you have to subtract 2 ATP. Minus 2 ATP is equal to 86. Okay? Okay. Hopefully that helps out.
So for the FADH plus one, or H, sorry, for the FADH2, we don't add inorganic phosphate?
You do.
We do, okay.
Yep. So that was already taken into account. If I think I understand what your question is, we don't add the 1 proton for the inorganic phosphate. You do. Right.
Okay.
Yep. So for this, when I calculated this, the 5 protons to make ATP, that's irregardless of whether those protons come from FADH2 or NADH. Right. So all we're looking at there is, okay, when we think about FADH2, is that can only pump 6 protons, whereas NADH can pump 10. Yeah.
Okay, cool. Thank you. Yeah, it does. Thank you.
Yeah, great. No problem. Other questions on that? Okay, so I got one in the chat. So we subtract 2 ATP for all beta oxidation, or does it differ by how long the chain is? For all of them, right? Whether it's a 4-carbon chain or a 28-carbon chain, okay, you would subtract just 2, because that fatty acid has to be made into an acyl-CoA. And it's only done once, irregardless of how long the chain is. Yeah, good question. Okay, good questions. I love the questions. Okay. Let's do one more question, depending on how long it takes. Okay, summary of each complex and its function. Okay, are the bolded answers on the practice exam true or false? I can answer that one right away. So the bolded answers are true. Okay, so I got that one. Okay, summary of each complex and its function. That'll take a little bit of time. Let's try and do it fairly quickly, though. I'll go ahead and erase this because I've run out of room on these. Oops. Okay. So I did this, I believe I did this in my last Q&A session, but I'll go ahead and do it again because I'm trying to remember if I recorded it. Apparently I didn't. But let me go ahead and do this real quickly. So when we look at complex 1, let's just do this. A summary of this. Complex 1 will take NADH and it will oxidize it to NAD+. Okay, and within this, there's going to be this FMN, a flavin nucleotide, and then I'm just going to put iron sulfur. There's like 8 of them that are involved in this whole process. And then there will be Q, and this is where we want to use a different color, Q. Right? So essentially the electrons go to this flavin nucleotide and then they hop up a bunch of iron sulfurs and they eventually make it to Q. And make QH2. Okay? Right. And all while pumping 4 protons from N to P side. So we'll put P over here, we'll put N over here. Okay. All right. Oops. Just a second. My dog is chewing on something, and I am worried about what he's chewing on. Just a second. Okay, never mind. It's a water bottle. He'll be all right. The water bottle is not going to be all right, though. Okay. Sorry. Some of you are like, I think I hear that in the background. It's this 1-year-old German Shepherd puppy. He chews on everything. All right, but anyway. All right, so that's complex 1. Now, complex 2. Complex 2 is really similar, especially when you draw it out this way, to complex 2 and to complex 1. But it's going to take succinate instead of NADH. And it's going to convert succinate, it's going to oxidize it to fumarate. Okay, and it's going to give, there will be another flavonucleotide, FAD, not FMN. And then there will be iron sulfur, there will be fewer of them, only 3 of them. And then that's going to have Q. It's going to be converted to QH2. Okay, so these electrons will go to FAD, hop, 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 up their way, and then finally make it up to QH2. Now, or Q, I put a H2, not QH2. There we go. Now this one doesn't pump any protons. Okay, so that's complex 2. Makes QH2 from succinate. Complex 1 makes QH2 from NADH and pumps 4 protons. Okay, now, this is where it can get wicked. Okay, so we'll kind of summarize this, but this is going to be the Q cycle. Complex 3 is kind of nutty in terms of how it gets its electrons through. Okay, and so what's going to happen is that those QH2 molecules, okay, are going to enter into the Q cycle. And because we're running out of time, I don't want to go over the whole Q cycle, okay? But let me just say this, okay? And this is a summation, and I'm not drawing it as much detail as this, all right? But those electrons are going to be passed through this to make 2 cytochrome Cs that will be reduced, and I'll just put RED, and you think, oh, they're red. No, reduce is what I mean by that, okay? 2 reduced cytochrome Cs, okay? And this is going to lead to the translocation of 4 protons. Okay, and I'll just say Q cycle, and that will explain how they get through there, okay? They'll go through, one of them will go from BL to BH to back to a Q and a Q inside, and then the other one was going to go to iron sulfur protein, cytochrome C1, into cytochrome C, and that's going to happen twice. Okay, so now after this, okay, we get to complex 4. Okay, and those cytochrome Cs will give up their electrons, and they're going to give up their electrons to copper A, and then there's a heme A and A3, and then copper A. And we didn't go over this in a whole lot of detail because we just kind of glazed over it. But essentially, these electrons make it through these things, okay, and they make it onto oxygen, making water. Now this is going to pump 2 protons. Okay. Right, so that's in total what we're saying. Now this comes back to our what we were doing last time. So NADH leads to 4 protons, 8 protons, 10 protons, right? Those electrons from NADH, you can trace it all the way through here. I'll use a big blue line. Those are the electrons from NADH. They make 6 protons, right? Right. If you were to look at then from succinate or FADH2, they are 4 plus 2 is 6. Okay, so that's where these things came from. Okay, so so that's a quick summary of what we spent several days on in class, of course. Okay, and that's probably all the time we have at this point in time. So so I'm going to go ahead and end it, turn off the recording, and I will hang out for a little bit. I can't hang out for